The biological stabilization of DNA nanostructures is essential for future in vivo applications, such as targeted drug delivery. However, DNA gets degraded in the body by various enzymes. Here we present a coating technique that protects DNA nanostructures from enzymatic degradation. The main advantages are that our coating comprises non-toxic compounds that are already used in living organisms and that the functionalization of the surface remains possible. This functionalization is important to incorporate molecular sensors for target recognition and switches for drug cargo release.
Polycation coating significantly increases the cellular uptake of encapsulated DNA origami structures. So this method can open the door for applications of DNA origamis in diagnostics and therapeutics. The cellular uptake is also required for potential gene delivery applications of the DNA nanotechnology. I consider that the method is quite straightforward and can be easily mastered.
To begin, measure the concentration of purified DNA origami. Use 2 microliters of TB buffer as the blank to calibrate ultraviolet spectrophotometer. And then measure DNA origami concentration at 260 nanometers. Then calculate the amounts of phosphates in the purified DNA origami solution using Formula 2 as described in the manuscript. Prepare 15 microliters of the DNA origami structure by including 1 nanomole of phosphate using TB buffer for dilution. Add 13.2 microliters of TB to 1.8 microliters of chitosan. And add 15 microliters of the chitosan solution to 15 microliters.